Thank you very much, Dr. Kubo, and thank you for pronouncing my name correctly, actually. So I would like to acknowledge the very nice invitation that I got from Sophian. Thank you for organizing this event. Thank you, Duncan, for all the practicals. And thank the advisory board for selecting me. This is a bit too strong, the voice, I think. And I have a confession to make. I'm not going to present any electrophysiological recording. I apologize for that. I've been in the ion channel field for 35 years. My first meeting was an ion channel meeting, so I'm very happy to be here. But you will understand from the object of my study that it's a bit difficult to do electrophy. So I'm going to talk about immune cells, and I'm going to first acknowledge the work of the two talented persons who did this. So my student Camille, she did the first study that I'm going to show, and Nicolas Rosa, a postdoc, who joined the lab 1 year ago, and if I have time, I will present some of his preliminary data. So the cell which is at the core of my interest is the neutrophil. So these are white blood cells. They're circulating in your body. They can leave the bloodstream to go explore the tissue, and their main job is to find and kill bacteria. It's not the only job that they do, but it's a critical one. If you deplete those cells, if you lose them, then you are really at risk of infection. They are the first line of defense. And the one thing which makes it complicated to study, they are very short-lived. Once they are in the bloodstream, they last there for a few hours, maybe 12, 15 hours. And they are highly motile, which makes it also complicated to study. And you can see in this slide the different steps that have been recognized by scientists and that are usually studied separately. This is in the bloodstream. They start rolling, and then they adhere. And then they can move on the vessel wall. They can cross. They can cross the barrier either through the cell or between cells. And inside the tissue, they can migrate, seek, find the bacteria, and destroy by phagocytosis. And I'm just going to show this movie, which was made using a new animal model that we have. It's the salsa mouse. It's a mouse that expresses a calcium-sensitive reporter in the white blood cells. And you can see these flashes are actually calcium elevation in the neutrophils during this phase of adhesion and crawling, which we mimic in a microscope using a flat substrate. So the other first event that we focused was this phagocytic event, the capture and killing of bacteria. And classically, scientists distinguish two phases here, the ingestion phase, during which the particle is internalized. It is to be recognized by surface receptor. These are either antibody which are decorating the particle or complement, okay? And then you have this phase of actin-driven engulfment, which is accompanied by calcium elevation, which you see here. When the cell turns green, the calcium is high, okay? So you can appreciate that pretty much every phagocytic event is associated with a global calcium elevation. But this is not over. When the particle is inside here, it has to be destroyed. So this happens by fusion of granules containing lytic enzymes with the phagosome, and the assembly of a very powerful enzymatic system, the NADPH oxidase. And some of those steps are also calcium dependent. And these are here local calcium elevation, which we visualize using a dye. In that case, it's the fluo 8 dye. So we focused on one class of channel, the store-operated channel. So this is made of two components. The channel itself is the ORI, calcium permeable channel, very selective for calcium. It's a plasma membrane channel, bona fide. But it's activated by another protein, which is an endoplasmic reticulum transmembrane protein. The name is STIM1. And this is actually the ligand of the channel. So it's a ligand-gated channel. But the ligand in that case is intracellular. It's a peculiar mechanism. The STIM1 is activated by the depletion of the endoplasmic reticulum, which occurs during signaling. Then you have a conformational change. The molecule extends and will then bind the plasma membrane via these domains and trap and gate the channel via another domain here, this groove, which then generates the calcium fluxes for signaling and refilling of the endoplasmic reticulum. So this channel was identified, it's now maybe 20 years ago, I guess, a bit less than that. And it was found in a family of patients suffering from severe combined immunodeficiency because it's essential for the differentiation of the T lymphocytes. So patients without functional OI or STIM, they suffer from severe combined immunodeficiency with infection in different organs. The other defect that is notable is a muscular defect. There are also some platelets issues and some other more specific effects. There are also gain of function mutation associated with mutation in either the STIM or the ORI. So we worked on some of those, and for that purpose, we do electrophysiology, and they are mostly characterized by muscular weakness. So this is the channel, which has a big role in the immune cells, mostly the T cells, but we studied also its role in the neutrophils. So a few years ago, Paula used this animal model, mouse lacking the STIM in all the myeloid lineage, and she looked at the implication of the STIM in the phagocytic event. So you see here this local calcium elevation, and I think you can appreciate here that the STIM protein is actually co-recruited at the site of calcium elevation in this cellular model of phagocytosis. And we characterize by electron microscopy the position, the recruitment of the STIM protein, which comes with its endoplasmic reticulum structure. And you see here a phagosome and here a structure cisternae of the ER. And you can see that if the cell line is lacking the STIM protein, this structure is smaller, so we quantify that. And the resulting defect is a defect of phagocytosis, which is about 50% reduction in the capacity of cells to take up particles. So we come up with this model, basically, that you need the STIM protein to bind to this ORI channel on the phagocytic membrane to generate these local elevation that are prophagocytic. They will favor the fusion of granules and the assembly of enzyme on the phagosome. So this is published in this series of papers. And now I'm going to start with an unpublished stuff, which is the work of Camille, using another animal model. So this is this mice which I presented, expressing the calcium reporter in the myeloid lineage. The reporter is two fluorescent moieties, a tomato, which is red, sorry, and the G-camp, which is a calcium-sensitive moiety. And what you see here is cells that are just flushed from the bone marrow of a mouse and that we adhere on polylysin. And if we do that, and you turn on the light, you can see that most of those cells are actually expressing the tomato, so they are myeloid cells, which is not unexpected, because the other one will not adhere. And if we now come and do a staining with a neutrophil surface marker, we can see that the majority of those red cells are neutrophils. So we now have a tool which enables us to study these cells in a one step, bypassing this purification step, which is time-consuming and depletes a lot of cells. So now we can just directly put the cells on a microscope, do the recording. At the end of the experiment, we label with the antibody, and then we can analyze the two populations, either the neutrophils or the other cells, which are mostly the monocytes. And if we now look at the calcium, just doing the ratio of green over red, and you see in this pseudocolor display that every time that there is a change in calcium, you see these cells turning green or red, okay? And you can see immediately that there is a very big spontaneous activity in these cells that we did nothing to the cell, no chemokine, no treatment, just adhere them to the substrate. Okay, so there is plenty of activity, and this, you can appreciate, looks almost like an ion channel recording. Okay, so every line is a cell, and pretty much all the neutrophil would, during the 3-minute recording period, exhibit at least one calcium elevation. So the first thing that she noticed was this: oh, there is a huge activity in these cells. I mean, this was sort of known. And this video here shows that using the same probe, you can also look in the turf plane, the adhesion plane. You can look at the calcium events, and you see that these are actually propagating calcium waves. So they will start here at the leading edge of the cell and then propagate towards the back. So we have a pretty good tool for high-resolution imaging here. I mean, this was known that the neutrophil will have calcium elevation, but the extent that we now observe is much more prominent than what was reported before. And this slide actually explains why. This is what you see with the Fura2 dye. When you load your cells with a dye, pretty much the whole cell is bright. With the SALSA probe, you see only the cytosol here. The nucleus, which is polylobed in these cells, is not loaded. So the probe is confined. And I think this gives us a much better dynamic. You see here in green, this is the SALSA. And the Fura would be like the violet. So you see that the buffering effect of the Fura is much larger than the one of the genetic probe. And we characterize this even further. And this is an activity. And this is a drug which blocks the phospholipase C. So this is a signaling dependent, so PLC dependent, nicely illustrated here. Immediate block of this activity. So there is signals involved. And then she turned to our STIM-deficient mice. So she crossed the STIM1 and STIM2. So this time, the two isoforms of the STIM are knocked down. She crossed it with the SALSA mice and measured the calcium elevation evoked by tabsikergin, which is the classical stimulus for store-operated entry. And you see, as expected, there is a big here abrogation of the response. Same goes for chemokine evoke response. And this movie here shows the spontaneous activity in the wild type and in the knockout. And I think you can appreciate that there is pretty much nothing. Camille told me I had to add tabsikergin at the end to prove that I am not cheating, that this is really a recording and not a fixed cell. So these cells express the probe. The probe is sensing calcium, but they are just not active. This is the graphical display, and the aggregate data are here. So the spontaneous activity is almost abrogated in these cells. And the few cells who still have activity still, this is also impacted. So what is the consequence of this defect? We observed here an incapacity of the cells to rapidly extend these lamellipods, so defect of spreading, adhesion and spreading. It's illustrated here. And we can record that also by turf imaging. So you see here the cell is coming on the plate and making this large adhesive plate. Another one is coming here. And at the same time, same speed, you see the knockout neutrophils are here. And if you quantify this formation of this adhesion plate, you see that there is a defect both in the speed and in the extent of spreading. So we have a need for the STIM protein for the neutrophils to spread. So at that point, I was convinced, okay, what we have here is defective STIMORI signaling, so we need to look at this contact sites forming, and I was expecting to see a massive defect in the knockout. And to our surprise, the knockout neutrophil not only had a normal amount of structure, they in fact had more in this adhesion membrane, which in retrospect is not so surprising, because to make this adhesion membrane, the cell has to generate this very thick actin cytoskeleton, which we believe is pushing away the contact site. So here you have the contact sites marked with those arrows, which are reduced in number in the wild-type neutrophil, contrary to our expectation. And can we quantify all those parameters, the proportion, and also the geometry of this structure? They are not altered. And in the end, the only, or one of the contribution which we could record was the basal calcium level. Between this oscillation, we noticed that it was also significantly lower in the knockout neutrophil, and we believe that this might actually impact the ability of the intracellular channels to release calcium during signaling. So to summarize these two stories, the first part, which is published, we showed, I think quite convincingly, that steam generates these contact sites at the phagosome, and that this is required for local elevation. But now the new finding from Camille show that these steam proteins actually are not needed for those contact sites or for tethering the ER structure, but they are required to drive this spreading and by promoting IP3-mediated calcium signal. And we're now exploring this hypothesis that we need the steam protein to send this receptor or to maintain them in a permissive configuration at the right location at the cellular cortex. Okay, so if now I have 3 or 4 minutes, I think I can go on the next story. And this is also preliminary, but we were prompted by this finding by Ardem, who got the Nobel Prize 3 years ago. He discovered these piezo mechanosensitive channels. Okay, so these are the mediator of touch and proprioception. Basically, these channels sense the membrane tension. And I figured that, oh, okay, maybe we need that also during phagocytosis in a neutrophil, and maybe not only during phagocytosis, because if you remember what those cells do, they deform a lot, okay? So our question now is, are these channels expressed in the white blood cells? Do they contribute to the calcium signals? Do they contribute to phagocytosis? This just shows you that during phagocytosis, you have a very big changes in membrane tension. So if you do have these channels, then you expect to see signals. So we first used the SALSA mouse model. And fortunately, Ardem developed a chemical activator of the piezo channel, which is this compound yoda. And you can see here that the yoda is generating quite a big calcium response when you add it. So it suggests that the channel is there. I showed this already. When the cell is taking up a particle, you see a big calcium elevation. And what we push the system by feeding the cells larger beads, 8 micron beads. Okay, so these are primary neutrophils for mouse. They can eat up to 3 of those particles. So they will go 1, 2, 3, and at some point, they cannot swallow more. But here, I think we are really pushing the system. And if there is a contribution of mechanochannel, we should see it. Okay. First hint that they may be implicated is using this toxin, GSMT4. But this is not specific for piezo. It's a toxin which, as I understand it, is blocking the membrane deformation. We do impact the calcium response in the neutrophils. So to get more direct evidence, we turn to genetics. So this is a piezo null cell line, which is a phagocytic cell line, human. It's PLB985 cells. And this is just documenting the loss of the piezo channel functionally. And this is how these cells behave when you feed them with 3 micron beads. This time it's fluo 8, but you see the calcium elevation, which is occurring every time the cell is eating. It's a complicated pattern, difficult to analyze. Remember, this is receptor-mediated events, so they are calcium elevation that are triggered by the receptor engagement. But if you compare the wild type and the knockout, and you integrate the response, you see that there is a significant reduction. So this suggests that there is a contribution of the piezo also in this cellular model. And if you look at the ability of these cells to phagocytose, it is reduced both in the number of cells that eat and the number of beads that they can eat. So then we turn to a more physiological system, which is the piezo null mice, so kindly provided by Eric Honoré, the University of Nice. So we crossed that with our myeloid reporter, got a nice depletion in the bone marrow and preserved expression in the lung. We got a reduction in the functional response. And I'm saving you the movie, but this is the 8-micron particles that are internalized by the wild type and the knockout neutrophils. And basically what Nicolas is doing is he's counting the holes in the cells. And this is the aggregate data. Pretty large impact, pretty large defect in the number of cells that eat and in the average number of particles that they can eat. And if you deconvolve that further, then you see that the knockouts failed to capture the third bit. So they are, after 2, they stop eating. So you would suggest you need the piezo-sensitive channel for this last step. So I'm done now. So in conclusion for this second part, I think we're convinced that the piezo-1 is expressed and functional in the myeloid cells, that if you remove this protein, this mechanosensitive channel, you impair phagocytosis, both in the human cell line and in the primary cells from mice, which leads us to conclude that the piezo mechanosensing might contribute to phagocytosis. And you may wonder whether this is relevant or not. And here is one paper that suggests that it might. In this study, they linked this piezo sensitivity to inflammasome activation. And they did one experiment which I had planned in my grant. So it's a bit of a bugger, but it's published like last December. So they used this animal in gout, mouse model of gout. So you inject uric acid crystal in the ankle, and you see that the piezo-deficient mice are protected. And we had reason to suspect that because one of the compounds, which in our hands works very well in blocking the piezo response, is this rainbow marron, which is known to be gout, an agent which is used to treat gout. So we think that the piezo channel is implicated in this phagocytosis of the crystals, and that if you block it, then you can prevent this gout. So I'm done, and again, would like to acknowledge a very great team, my senior fellow Amado, and the 2 persons who contributed, Camille and Nicolas. And I'm happy to take questions. In the neutrophil, I cannot answer, but we have this Western blot, and if you take the whole myeloid compartment, then I think you can definitely see it. No, compared to lung, and first of all, it's not the same weight. I don't know why, but this is a huge protein. It's a trimer of, 52 transmembrane domains, or maybe some glycosylation happening here. So it's definitely there. That's all I can say at this stage. And you can see it functionally also. You know, I mean, you guys are ion channel people, so you don't need a lot of ion channels to make a functional response.
No, for sure. But we know that, when recording primary cells, it can also be difficult depending on the expression level, of course.
No, the neutrophil, as a population, they are very diverse. So I don't think everyone may have it. But if you have like even 50% or even 30% of neutrophil will express it, that would probably be sufficient to give you a functional response at the animal level.
Okay, thank you.
Any other question? Yes.
Thank you for your talk. Is the idea then that STIM1 and piezo would be the 2 main sources of calcium influx in these cells?
No, I would not go that far, but these are the 2 main components that I'm interested in.
Okay. But what other components would there be?
There are quite many papers on TRPM7 in these cells. I mean, there's also TRPC3, which has been reported. So there are, like I said, the neutrophil, it's a population. If you take them as a population, they express pretty much the whole genome. So you can publish whatever you want on this cell. The key question is, what's the proportion of cells that would express it, and how would it contribute to the global behavior? It's a bit like an army that you give different weapons to your soldiers.
Okay, thank you.
Okay, one last question. Yes.
Thank you very much for a nice talk. In your study, you used bees coated with IgG, but that's good for separating between early steps and following steps. But if you use like more native substance such as zymosan or even pathogen, the outcomes are different, or do you still see the same findings?
Yeah, good question. So we did use zymosan. I was too fast in the cell line, which is here. This is actually zymosan.
Oh, okay.
And this is complement mediated. So if you don't coat, they will not eat. So that's a bit of a hurdle that you need to put something appetizing on a bead, and then you engage the receptor, and then you have calcium signaling because of the receptor engagement. But it works both for the IgG coated and for the complement coated zymosan. And of course, we have plant pathogen, so that's the next step. The one thing I like about the uric acid and the gout is that those crystals, they would be eaten without opsonization. I think the cell will at least try to eat that. So that's maybe pure mechano sensation.
Yeah. Thank you very much.
Okay, time is up, and I'd like to thank Nicolas again.